What we've been pushing is a cultural change in how people do science.
We can't expect that everything would be absolutely reproducible.
What makes me angry is that some people in the community thinks that this is easy.
Science is hard and collecting data is hard, and so there's a lot of places where you can have flaws.
Science is defined by the accumulation of knowledge over time through the systematic observation and gathering of empirical evidence. In the life sciences, as in other scientific disciplines, each new finding is built upon a foundation of generally accepted facts that have been vetted by researchers and disseminated in the literature for other scientists to reproduce. But what if the majority of the facts published in the literature are not reproducible? What would that mean about our state of knowledge in the life sciences, and how would it impact our search for the cures to diseases? The issue of reproducibility in science has gained a lot of attention over the past few years, and for good reason. The very foundation of how biomedical research is performed is being called into question. Currently, the workflow for science has involved the close interactions of 6 different stakeholder groups. Researchers, who are responsible for formulating a hypothesis and designing experiments to test it. Suppliers, who provide reagents and equipment to help perform those experiments. Professional societies that foster collaborations and help disseminate information amongst researchers through conferences and posters. Publishers, who are responsible for assigning reviewers who scrutinize the research to decide if it is worthy of entering into the written record. Funding agencies that prioritize which research is worth pursuing by granting scientists the resources to continue their work, and institutions, both in the private and the public sector, where this research is taking place. These stakeholder groups together form the life science community. But the pace of discovery for this entire enterprise is dramatically affected if a large percentage of the research that is disseminated is not reproducible. So how big of a problem is the issue of scientific reproducibility? And what, if anything, can be done to address it? Here we will take a deep dive into the problem of reproducibility in basic research to examine some of the root causes. In particular, we will focus on the role of one ubiquitous reagent that has been implicated by many as a significant contributor to irreproducibility: antibodies. In recent years, these powerful, highly versatile proteins have been proven to be extremely effective as therapies for a wide range of diseases. But their use as a research tool goes back for decades, and they have helped researchers push back the boundaries of science. But their widespread use and misuse in research labs may be contributing to the irreproducibility issues the life science community is currently facing. We will talk with representatives from key stakeholders in the life science community to find out what they think are the problems and the possible solutions to the reproducibility crisis.
The most important thing to realize about this whole set of issues is that no one stakeholder group can solve this. NIH alone can't solve it, investigators alone can't solve it, journals alone can't solve it, institutions alone can't solve it. We're all in this together.
The current focus on reproducibility in science can in many ways be traced back to an article written by Glenn Begley and Lee Ellis that was published in the journal Nature in March of 2012.
I didn't start out with the intention of stepping into this mess. What I started out to do was, when I was at Amgen, was to prepare for my successor. So one of the things that we did early on was create project reviews.
The ability of his group to reproduce 53 seminal studies in cancer, they were only able to reproduce the seminal findings. That's the important point. Not every data point, and you don't decrease tumor growth by exactly the same amount, in every study. But the seminal findings should be reproducible, and his group was only able to reproduce it in 6 out of 53 studies.
When Begley and Ellis did their study, it was sort of in the 15 to 20% range of reproducibility, and then subsequent other studies that have been done that are sort of in the 20 to 25% reproducibility overall. And these sorts of studies suggest that that's sort of where we are at the reproducibility issue, or somewhere between, at best, half of the studies are reproducible and maybe less than 25%. What causes that lack of reproducibility isn't really so well defined, and it probably represents a lot of factors.
In a paper published in PLOS Biology in June of 2015, Len Friedman and colleagues attempted to identify the potential sources of irreproducibility in the life sciences and associate a dollar figure to each source.
You know, in conversations, we talk about what the financial impact of this would be, what the economic impact of this would be. And in one of our initial events, we decided to try to do an analysis for irreproducibility. We calculated the amount of spending which is publicly available on both clinical and preclinical research and divided that by a 50% irreproducibility rate, and we came up with this $28 billion value. Needless to say, that definitely rustled some jimmies, if you will.
In his paper, Friedman outlines 4 areas that can contribute to issues with a study's reproducibility. These include laboratory protocols, data analysis and reporting, study design, and biological reagents and reference materials. This last category itself is made up of several different types of reagents, including animal models, cell lines, antibodies, and other reagents. Our focus will be on antibodies.
Informal conversations I've had with people like Glenn Begley, he will tell you that a big source of the inability to replicate the papers that they tried to replicate were, one way or another, the source of the antibodies.
We were interested in trying to develop a diagnostic test for melanoma, and the idea is that our diagnostic test would detect those patients that were likely to have their melanoma recur. We built and did a training set and a validation set and showed that we could tell whether a patient had about a 7 or 10% chance of recurrence versus a 40 or 50% chance of recurrence just on the basis of these tests, and we published that back in 2009, and then we subsequently got a second R01 to validate the test and begin to prospectively test it in the commercial setting or in the clinical setting. The key reagents in the test were 5 different antibodies, and of the 5 antibodies, 2 of them were completely non-reproducible. That is, we bought a lot of antibody that had the same number from the same vendor. We never published a follow-up paper because we couldn't reproduce the signature and thus couldn't reproduce the clinical test.
The problem that the RIM lab faced was most likely the result of antibodies that had not been properly validated. The issue of antibody validation is currently an area of great focus for the companies that manufacture and distribute antibodies to the research community. During the 1980s and early 1990s, antibodies were primarily generated by academic research labs. This meant that the creation of a new antibody was both time and labor-intensive, and the number of antibodies to any given antigen was limited. The 1990s saw a growth in the number of antibody suppliers, fueled in part by the Human Genome Project. As new genes were identified, tools to work with those gene products became necessary. The benefits of the groundwork laid down by basic research in the 70s and 80s, as well as the initial work on the Human Genome Project, soon became apparent as the number of FDA-approved antibody-based therapies began to grow in the mid-1990s. Today, biologics, as these therapies have come to be known, represent some of the most exciting advances in medicine as they extend our arsenal of treatments for diseases such as cancer, autoimmune disorders, infectious agents, and even inflammatory and neurological conditions. During this same period of time, the use of antibodies in the research labs expanded dramatically. A search on PubMed for publications containing the term Western blot in 1990 yields 2,949 results. By the end of the decade, that number had more than doubled, and by 2015, there are almost 10 times the number of Western blot publications in PubMed than there were in 1990. A similar analysis for other antibody-based techniques yields similar results. At the beginning of the 1990s, there were approximately 50 antibody suppliers that sold around 13,000 antibodies. Today, the antibody market consists of 350 to 500 suppliers that sell more than 2 million antibodies.
Before the Human Genome Project, there was several thousand antibodies, maybe 10,000 antibodies, and literally within the course of 10 to 15 years, you had hundreds of thousands, if not millions, of new antibodies entering into the market. And so I think what that's done is it's created an accelerated need to validate those antibodies better. And in order to really validate an antibody, you have to understand the biology of the target.
When we talk about validating an antibody, what we're really talking about are 3 properties. The first property is sensitivity, or the ability of the antibody to recognize the intended target. The second property is antibody specificity, the ability of the antibody not to recognize things that it should not, in fact, recognize. And the third property, which is equally as important, is reproducibility. we should be able to demonstrate to the end user that there is reproducibility, not only as you repeat a specific experiment, but on a lot-to-lot basis.
As the number of companies that sell antibodies grew, it soon became clear that not all antibody suppliers were spending the same amount of time and effort to validate their antibodies. While the large number of antibodies in this market means it's easier for a researcher to find an antibody to any given antigen, there are issues of transparency and product quality that can make it difficult for them to know exactly what they are buying. This was in part because as the number of antibody providers grew, the way their products reached the market became more complex. It was no longer the case that the company that produced the antibody sold it directly to researchers. This has led to a call from the research community for more clarity about the original source of any given antibody.
There are many paths for research reagents like antibodies to be delivered to research scientists. There are companies that produce materials that also sell them. There are distributors that distribute products from many companies. And there are companies that purchase and resell products from manufacturers, but when they resell them, they sell them under their own brand name. That's called the OEM path to the marketplace. The most important thing is the aspect of transparency. If they have all of that information that tells you how the antibody was made and tested, they're going to be able to reproduce their studies regardless of the path that the reagent took to get to them.
It may not be realistic for companies to disclose the origin of the antibody. It could be that there are contractual agreements with the supplier that prevents companies from actually disclosing that. However, this is more of a problem for polyclonals because it would be possible to really track where they come from. But in terms of the monoclonals, it's quite easy to say that this is the same antibody because you have the clone number.
It's really tricky because quite a few of our suppliers are not prepared to go to market directly. So we're basically their go-to-market channel, so to speak. So suddenly saying this comes from this supplier, it's transparent, but it's not really, that supplier's not prepared to deal with the technical issues, deal with customer calls, ship things all over the world. And the other point is many of us are adding lots of data and doing a lot of work on our own side. It doesn't address the value add that we're putting into products, whether we're doing CRISPR knockout, for instance, across many suppliers' products.
While there are a wide variety of ways for an antibody to come to market, one of the questions that some think is equally or even more important is the type of antibody that is being sold. The vast majority of antibodies sold today are either polyclonal or monoclonal, but advances in cloning and expression technologies have made the generation of recombinant antibodies possible. Recombinant antibodies are molecular clones of a given antibody that is expressed in a system such as the CHO cell line. In a Nature commentary published in February of 2015, Andrew Bradbury and Andreas Pluchthun called for the entire life science research community to abandon polyclonal and monoclonal antibodies in favor of recombinant antibodies.
The fundamental issue about recombinants compared to all other antibodies was their definability. And this is something that you don't see with monoclonals or with polyclonals. They're not defined at the molecular level.
Interesting example that I've recently been involved in is an antibody that has been in the research world for quite a number of years. It came out of an academic lab. The hybridoma was made publicly available, so a number of companies are marketing it. And we recently needed to engineer that antibody, so we cloned it out based on a sequence that we were able to get from a patent. And when we cloned it, we realized that the hybridoma material that we'd been using was a mixture of two antibodies. And only one of those antibodies is a high affinity antibody that is advertised to do what we all think it should be doing. You could argue that's not a problem because everybody's been using the same thing, but what we don't know is, has that mixture changed from lot to lot? Has it changed from year to year? And could contribute certainly to working well in one group's hands and not in another.
The hybridomas, they cannot be adapted to growth in medium without FCS. Many of them actually expressed additional light chains from the original fusion partner, from the myeloma. And that can vary from batch to batch. At the end of the day, you purify an antibody, but it's actually more than one antibody species. It's maybe one heavy chain, but 2 light chains. And the mixtures may vary from batch to batch. That is actually a nightmare. You can much more standardize processes with recombinant technology, and that is not invented by us. That is known in the field. Yeah. People in the therapeutic business, they know that.
You can work with hybridoma material and polyclonals and characterize it to a degree that you can have the confidence that you're making the same material every time. So it has the same amount of non-aggregated material, the same amount of clipping, that has the same behavior. So if it's a polyclonal and you characterize it for immunoprecipitation and lot after lot it does the same immunoprecipitation, do you need to clone it out recombinantly? Because that's actually quite a lot of work. And to then prove that you have the right antibody and prove that you're making the right antibody is a significant endeavor. And so there has to be a larger picture driver for why you would want to do that.
While the source of a given antibody, as well as the quality of that antibody, can play significant roles in reproducibility, these are not the only issues. One topic that is often brought up by all stakeholder groups is training. Many researchers do not understand the underlying complexities of using antibodies and how to avoid issues that could yield false results.
One of the solutions to the overall issue around reproducibility is better training for students, postdocs, even established investigators.
Researchers, particularly graduate students and postdocs, really need to start thinking about the tools that they're working with. Antibodies, unfortunately, we take for granted what someone says it is, and we may overassume what it's capable of because we don't actually understand what we're working with. And antibodies definitely fall into that category.
The end users need to follow the instructions of the protocols that are provided by manufacturers to use that antibody and get the right result. And they need to be good scientists. They need to do the right experiment. They need to add controls and everything to make sure that everything is working properly, which is back to training and education. So I think that there is a little bit of a lag in how perhaps the younger scientists are educated.
The reason why we have reproducibility problems, bad reagents in some cases, but I also think that one has to be very careful to see that you don't have off-target in your assay, and that of course means that you need training and a better understanding of how difficult it is to use antibodies.
Reviewers there's not one reviewer who is careful about looking at the antibodies, who's sort of expert at that. That first high-impact paper is really important, and then everybody kind of follows it. And if that's not done well, or if the materials for that aren't qualified well, then the whole field downstream develops on the basis of a poorly qualified material.
The difficulty of reproducing data from the literature has also come up from the different stakeholder groups. One question that has been posed is, do publishers demand enough details in the manuscript to allow for the work to be replicated? As the gatekeepers of the written record, many see the publishing community not doing enough to ensure that everything that is published is reproducible.
One of the top journals, not too long ago, before Asilomar, I went to a paper because I was interested in the paper. And then I went to go and see which antibodies they use. There was no information, none whatsoever. So that paper is irreproducible.
One problem I believe in the reproducibility issue that we have is that journal articles do not contain enough information to allow me to reproduce your work. How often might I have to go and call a researcher and find out after I've tried it 2 or 3 times? What's the magic sauce that made your work look so beautiful so that I can replicate it in my own hands?
Most papers in the scientific literature simply do not give enough information so that you can track down these antibodies very easily. For example, an author might say, I used the GFAP antibody from Sigma, and it was monoclonal, and here's my dilution. Well, when I go to Sigma, I would see that there are 40 antibodies for GFAP, and I don't know which one they used.
The most important responsibility that we have, I think, is making sure that the experiments are described with enough detail that they can be reproduced by someone else who wants to replicate the experiments or extend the work. And we've increased the amount of supplementary information that is made available and increased the scrutiny both internally and from reviewers to that supplementary material to make sure that it looks like everything that's needed to be there is there. But with that said, I think there's still plenty of room for further enhancements. There are hundreds of different steps, and there's a balance between describing everything in complete detail, but making it completely unreadable.
By putting too much emphasis on the publishers, you really are catching things very late in the process. Researchers don't have this validation in a format that they can easily pull out and provide at the time of reporting. So it needs to be a continuum, right? It needs to be something that researchers think about when they write their grant application, when they conduct their research, and when they report their research in an article. And I think that we don't necessarily have this continuum at this point.
The reproducibility crisis has galvanized organizations such as ABRF, FASEB, AACR, SFN, ASM, and others into action. In 2014, one group, the International Working Group on Antibody Validation, was founded with the purpose of developing standards that could be applied for the validation of antibodies in an application-specific manner.
Back then in 2014, we decided to form an international working group for antibody validation. Scientists from North America, from Europe, and from Asia came together during 1 year, and we discussed how to best have formal validation of antibodies. And we then published a paper in October 2016 in Nature Methods, where we are proposing 5 formal, we call them pillars, for antibody validation. They are independent of any prior knowledge of the target, and you can use them in an application-specific manner. And one of the things that we really push in this proposal is that all validation of antibodies has to be done in an application-specific manner. And then what we're also adding then, if you can, try to formally validate them through one of these 5 pillars.
About 2 years ago, we actually provided the funding support to form the IWGAV and to provide, basically support, financial support, so those folks could get together and really tell us what they felt like what was going to be the most impactful thing that we could do. I believe they identified some really well thought out techniques. We're going to take all of the efforts of the research community, including IWGAV and GPSI, and the feedback we felt like that we've gotten to move our standards going forward as well.
In the fall of 2016, a conference was organized by the Global Biological Standards Institute in Asilomar, California, to follow up on the groundwork laid by the IWGAV. This meeting included representatives from all stakeholder groups in the life science community.
Getting all the stakeholders together at a small venue like Asilomar, came about. We were approached by Jim Houston, who was the president at the time of the Antibody Society, and we started working together on planning this meeting. It was actually Jim's idea to do the meeting at Asilomar, given the sort of historical panache, if you will, that Asilomar has. And it was going to be critical to have all the stakeholders there. And I think in that respect, we were very successful. I began to have conversations with all of them, including Matthias Uhlen. The pillars would be part of the discussion at Asilomar. And the pillars sort of created a foundation for discussion. The other component that was discussed and really sort of took on a life of its own at Asilomar is the scoring component. It seemed to really get some traction at the meeting.
I think it would be great to have a means to determine whether an antibody is good. I've tested it in this application, and I can rate the sensitivity in some way, I can rate the specificity in some way. And I think a lot of this will be predicated on having the data publicly available too.
The idea is that not only will the scores be published in some central kind of way, but the data behind the scores will all be referenced. So you can go check and find out where it is, whose website it's on, so that you can be your own judge in terms of how well validated the antibodies are on the basis of a scoring system.
While there may be excitement among some in the research community about the prospect of an antibody scoring system, there are still details with effectively implementing such a system that need to be worked out.
There's a very strong push by the GBSI to do this scoring. I am personally very skeptical about it, and I was vocal about it, and I keep being vocal about it. I mean, I'm in a working group now, the IFC working group, that is pushing to a scoring system. And honestly, I don't think it's ever going to work. Who is going to enforce them? Who is going to determine the score? Who is going to review it? Are the people who are going to do that, are customers going to care about that scoring? And I can tell you that the industry is skeptic as a whole on the scoring system, because it's not very clear who will be the person who scores, who will have the money to really constantly do these things.
I think the scoring system is going to be complicated. I think when we put all the pieces in place, we will see that it might be more complicated than we thought originally.
We're hoping that the first drafts of all the working groups' work will be completed by the latest June. The plan is to then put it out for public comment, and then a very detailed manuscript would be written and submitted for publication by the fall. We would then start working very hard on implementing.
Antibody suppliers have begun to actively engage with researchers and societies to address reproducibility issues. Many are currently engaged with the working groups that arose from the Asilomar meeting to help bring the application-specific validation methods to the market. But even as this work is going on, there are some immediate steps that suppliers can embrace when it comes to data transparency. If researchers can see both the positive and negative validation data that many companies are currently generating before releasing an antibody, they should be able to make smarter purchasing and usage decisions.
The discussion that we're having right now internally is how can we perhaps house negative data collectively, but I think even more specifically for us is we're trying to figure out how we do it distinctly on our website, for example, so it doesn't commingle with the data and provide confusion.
So a lot of work, actually the past 3 years since I've taken over a leadership role, has been put into making sure that we have an image to back every single claim. It's a lot of work to do that, and we know that they work, but we didn't actually always have the images up on a website. So making them publicly available and on the data sheet has been a big priority. And I'd say half of our efforts right now are put into adding more application data.
The transparency only comes under the pressure of both worlds, the scientific research world and the manufacturer world. Don't think you're just producing a publication. When you provide medicine to a patient, don't you want to control what is going into your process? If you know that, if you have that kind of responsibility on your shoulder, you will be very, very adamant to know the exact origin of your supply. A lot of pharmaceutical company has demanded the supplier to tell what is a manufacturer. And they even need to know all the manufacturer data from the process of the particular product. So ask for transparency, and that is a solution for now.
Funding agencies such as the NIH have already begun to implement changes to address reproducibility. But the question of what role the research institutions can play remains to be seen.
We've sort of revamped how we do review of our grant applications. If part of your supporting information comes from somebody else's laboratory, typically what we do is we just allow the applicant to cite the work. Well, what if that published work of another group had some flaw that was inherent? And so part of this new review approach is to look at the premise of the application, which ensures that not only the work antecedent to the application done by the investigator is reviewed, but also a quick look is taken at what the work that is cited from other groups is about. Second element is, what are the methodological approaches in terms of experimental design? And are the basic principles of experimental design adhered to? The third ought to consider the right variable. Has the applicant explained why he or she is only looking at one sex of say, an animal model? Or have they made the case that they are going to be looking at both sexes? And then finally, there's the idea about validation. Have you appropriately validated all of the materials that you propose to use in your grant application?
There's one group that's being derelict in their responsibility, and that's the institutions, the universities, the research institutes. They, to my mind, fail to take responsibility for the investigators that they house. the universities, the institutions, are not doing enough to ensure quality coming out from their walls, from the work that takes place within their walls. Each institution could have an Office of Scientific Transparency or an Office of Scientific Reproducibility or something along those lines, and that would be the office that is charged with ensuring that if I publish a paper in Nature, my institution checks that the data is actually available, that when they go through the lab notebooks, if the experiment was done in a blinded fashion, it's recorded there in the lab notebooks, that I didn't p-hack and find the result that I liked best for publication, and so on. It's too much for the NIH to have those sorts of controls, but it is appropriate, in my view, that the institutions introduce safeguards along those sorts of lines.
As publishing moves away from the printed page towards the internet, the ability of the publishing community to address reproducibility issues grows. This includes ensuring that more detailed information is included with every published paper. It could also include the adoption of new ways to convey information, such as the adoption of preprints, as well as other ways to communicate both positive and negative data.
We need to be able to identify unambiguously which reagents are being used. That requires reporting of the reagents. You know, it's not only the vendor, it's the catalog number, it's the lot number. I think the more granular we can go into the identification is really the first step that we need to have.
We've been working on the Resource Identification Initiative, which is an initiative that started with about 25 journal editors-in-chief. Over a couple of years, we sort of came together, we tried to figure out what would be the best ways to improve reproducibility in the journals that they were heading. And then what we did is we asked authors to essentially put unique identifiers into the papers from these 25 journals. By the end of the first year, we had those 25 journals. By the end of the second year, we actually ended up with 50 journals. Right now, we're over 250. There's a whole lot of journals now really coming on.
You know, preprints, the preprint server movement, which has been taking hold and NIH just sort of embraced, is a good vehicle, I think, for people to share negative results that, you know, they won't be peer-reviewed, but they will be out there.
So our view, and I'm talking about Bruce Alberts and I here, was really not to try to punish people for making mistakes, but rather to make it easier for the disconfirming information, you know, the replication studies to be published. It seems to me that the internet makes that very easy. And obviously there's still some mischief that can happen through the internet, but I guess I was struck by how good really Wikipedia is, for instance. It seemed to me that if there was a little bit of an opportunity and a culture to publish the data you had, and I'm not talking about just a line that says that paper is crap, you know, I'm talking about a mature, data-rich publication where you don't even necessarily have to say, hey, this is wrong. You just say, you know, this is what we did, here are our data, here are the controls. Try to level the field a little bit so the work that is not supportive of a high-profile publication gets out there more easily.
While each stakeholder group in the life science community views the issue of scientific reproducibility through a slightly different lens, there is hope that these different viewpoints are beginning to focus and bring light to solutions that are meaningful, fair, implementable, and flexible enough to revise.
In 2012, we received hate mail and were abused and so on. That has changed completely. In 2016 and 17, if I give a seminar on the same subject, there is almost never anyone that says, that can't be true, I disagree with you. That just doesn't happen now. When I talk particularly to young scientists, they are very keen to address this issue.
Maybe a year from now, we'll be talking about the beginnings of scoring systems being published and people actually using scoring for antibodies. Maybe 2 or 3 years from now, seeing antibody companies actually advertising the quality of their antibodies on the basis of these scores. I'll have a pretty good chance that what's on that tube is what's in the bottle.
I think the most important thing is education. The training is the most important thing. Having an astute scientist at the bench knowing what to use, what to buy, where to buy it from, how to use it properly, that is 90% of the job, I think.
a community effort is going to come together and we are going to see a change. The reason I believe that is some of the great things that are happening in science today. Antibodies are critically important reagents, and if they're used correctly, if the antibodies are produced by manufacturers in such a way that they're well validated, if the research scientist uses them as fit-for-purpose reagents, that is, that they use them in a way that they were intended to be used when they were made, we can do great things. We can collect great data. We can solve yet unsolved problems.
These things are going to take time, but I do see movement. I do see positive movement because it has really forced researchers to be aware that it's important. They are now a lot more conscious of it. They can't really ignore it. And if they can't ignore it, then they're going to have to address it. That's only in the last couple of years. So I think if you're looking at, 5 years or 10 years, if the pressure continues to build, and I think it probably will, then I think that there will be sort of a cultural change here.